Imagine redesigning life at its most fundamental level. Scientists in the UK have taken an astonishing leap toward this by engineering a synthetic version of the bacterium Escherichia coli, commonly known as E. coli, armed with a drastically simplified genetic code. This bold achievement involved using only 57 of the original 64 codons, the triplets of nucleotides that serve as the language of life within DNA and RNA. But here's where it gets controversial. Could such a reduction in genetic diversity limit life's potential, or rather, could it unlock new possibilities? To understand the significance, let's break down what codons are and why they matter. Our genetic code is composed of 64 unique codon triplets, which are sequences of 3 nucleotides. These triplets provide instructions for cells to build proteins by specifying which amino acids to add and when to halt the process. Proteins, made from 20 common amino acids, are essential players in virtually every biological function. Interestingly, because there are more codons, 64, than amino acids, 20, many codons are synonyms, different codons that instruct the cell to add the same amino acid. The team behind this synthetic E. coli strain, referred to as SYN57, decided to remove some of these seemingly redundant codons, compressing the genome to a minimalistic but functional set. The method was painstaking. The scientists chose to eliminate 4 out of the 6 codons coding for serine, 2 of 4 codons for alanine, and 1 of the stop codons, substituting any occurrences of these removed codons in the bacterial genome with alternative synonymous codons that provide identical instructions. This monumental rewriting required over 101,000 individual genetic edits. Initially, these edits were designed in silico, divided into manageable segments about 100 kilobases in size. Following this digital blueprint, the team progressively assembled the genome piece by piece, continuously testing fragments inside live bacteria to ensure no fatal errors were introduced. Ultimately, these fragments were stitched together to produce a fully synthetic bacterial strain with a streamlined genetic code. This accomplishment is a milestone, proving that life can survive even with a significantly condensed genetic code. But the implications go far beyond mere simplification. By freeing up codons, this approach opens the door for scientists to encode new, non-standard amino acids, potentially leading to the creation of novel proteins with unprecedented properties. As the researchers from the Medical Research Council's Laboratory of Molecular Biology explain, SYN57 could allow the development of innovative synthetic polymers and macrocycles, materials that might revolutionize fields from medicine to manufacturing. Now, here's the point most people miss. The rewritten genetic code of SYN57 is effectively incomprehensible to natural microbes, including harmful viruses that typically sabotage bacterial protein production. This natural resistance to infection is a game-changer for industries relying on bacterial cultures, where virus contamination often causes massive losses. Moreover, this unique genomic makeup acts as a biological safety lock. Genetically modified bacteria with such a synthetic genome are less likely to transfer their engineered genes to natural ecosystems, addressing one of the biggest environmental concerns about biotechnology. While touted as groundbreaking, this research also invites debate. Does compressing the genetic code risk reducing biodiversity or adaptability, or is it a necessary step toward designing life forms tailored for specific applications? The study's findings, published in the prestigious journal Science, pave the way for intense discussions on synthetic biology's future and its ethical boundaries. What do you think? Is minimizing the genetic code the smartest path forward for synthetic life? Or are we tampering with nature in ways we don't fully understand yet? Share your thoughts and join the conversation.